In the process of recombinant DNA technology, polymerase chain reaction is one of the most important steps. In polymerase chain reaction, a DNA fragment can be multiplied into several copies within minutes or hours. So actually, PCR basically means DNA replication on large scale. This technique was developed by Kary-Mullis in the year 1980. PCR requires the following. First, number one, PCR machine where this entire process is carried out. Next, a fragment of DNA which we are calling it as our gene of interest, or desired DNA, which acts as initial template. Then we require several deoxynucleotide triphosphates which acts as substrates for our DNA polymerase. So all the 4 dntps we need, like adenine, thymine, guanine, cytosine, all the 4. Next, DNA polymerase is an enzyme. Here we are using Taq polymerase. This is isolated from Thermus aquaticus. Because enzymes are very sensitive, here this entire process of PCR is a temperature dependent process. So Taq polymerase can withstand to a temperature of around 95 degrees. So we are using Taq polymerase enzyme in this process. Next, primers. A pair of primers with exposed 3' OH ends. Right? So these primers are required which will add to the nucleotide sequence present on the template. So the question comes, why primers are required? Always DNA polymerase add nucleotides from 3' OH group of the growing strand. So as a result, primers are required to initiate the process of replication. So for that we need primers. Next, this entire PCR include 3 temperature dependent steps. First one is denaturation. Second one is called Unlean. Third one is primer extinction. So first, denaturation. Initial mixture is heated to 94 degrees. At this high temperature, the hydrogen bonds between the 2 strands of the double helix break, separating into 2 single-stranded templates. This is followed by second step, which is unleaned. The reaction mixture after a couple of minutes is cooled down to 50 to 60 degrees. In different books it is mentioned with different temperatures like 50 to 54 and so on. So we are taking it between 50 to 60 degrees. At this temperature, primer identify a complementary sequence of nucleotides on the template and add to the template. Right? So mostly primers add at the 3' end of the template. Then followed by the third step, which is primer extinction. In the primer extinction, Taq polymerase, that's our DNA polymerase, add nucleotides to the primer which are complementary to the template strand so that the chain will grow, synthesizing a new strand. Always the new strand is synthesized from 5' to 3' direction. So these are the 3 events: denaturation, unlean, primer extinction. So if this process is repeated of the several cycles, so DNA fragments can be produced to billions copies. It is possible to generate 2n number of copies after n number of cycles. For example, if you are taking a single DNA fragment, if you are going for around 4 cycles, so 2 multiplied by 4, that means you will get 16 copies at the end. So this is how the polymerase chain reaction is used for multiplying the gene fragment.